Okay, hello everyone. I'll give a closer look now at this paper from Royal Society from 2008 and how much of protein sequence space has been explored by life on Earth. We conclude that rather than life having explored only an infinitesimally small part of sequence space in the last 4 billion years, it is instead quite plausible for all of functional protein sequence space to have been explored. Sequence space is the number of possible combinations given a protein with a specific size or number or amino acids. So as an example, if we want to calculate the number of possible combinations of a protein with 100 amino acids, since on each position there can be selected from 20 different amino acids, the sequence space is 20 to the 100th power, or 10 to the 130th power. Now let's make a quick calculation. There have been roughly 10 to the 16th power of seconds since the Big Bang. 10 to the 43rd power is the maximal number of possible interactions of atoms with each other in 1 second. There are about 10 to the 80th power of atoms in the known universe. 10 to the 139th power is the number of possible events to occur in the entire history of the universe, calculated multiplying the number of atoms with seconds since the Big Bang and the number of possible interactions of one atom with another per second. The computer scientist Seth Lloyd from MIT calculated that the maximum number of bit operations that the universe could have performed in its history, assuming the entire universe would have been employed in its entire history, all its efforts just to this task, is 10 to the 120th power, meaning that a specific bit operation with an improbability significantly greater than 1 chance in 10 to the 120th power will likely never occur by chance. So he actually is a little bit more gracious, let's say, in regards of that number. Now, if we calculate the probability of producing a single functional protein with a chain of 100 amino acids by chance, it is 10 to the 130th power. That means the shuffling during the history of the entire universe for 13.7 billion years, the cosmic lottery would have exhausted the possibilities to find the right sequence just for one protein with 100 amino acids amongst 10 to the 130th power of different possible combinations. We can read about this at Steve Meyer's book Signature in the Cell in the 10th chapter. So Pelagibacter Ubique is one of the smallest free living bacteria known today. It has a small genome of about 1.3 million nucleotides, which encode for about 1,350 proteins. The average protein size of bacteria is about 320 amino acids. That means just to have one protein with an average size of 320 amino acids, that means exhausting the probabilistic resources of the entire universe to get that protein by random events and a trillion times. Now remember that the shuffling would have to be done 1,350 times to have the entire protein set used in Pelagibacter Ubique. But this bacterium supposedly evolved from an alpha proteobacterium, a slightly larger common ancestor with about 2,000 genes. And the estimation is that alpha proteobacteria arose about 1.9 billion years ago. And the oldest known forms of life are supposed to be much older, up to 3.7 billion years, evidenced by formations called of Stromalotites. Stromalotites are the product of photosynthetic bacteria. And the smallest described genome of a clade of cyanobacteria is Raphidiopsis and has about 3.2 million nucleotides. And its genome encodes for 3,000 proteins, so it's significantly bigger. So this is the paradox. Raphidiopsis, the smallest cyanobacteria, belonging to the oldest life forms on Earth, has about 3,000 proteins, while Pelagibacter Ubique, currently the smallest bacteria, has just about 1,350 proteins and emerged supposedly 1.6 billion years later. If we apply evolutionary thinking, should it not be the inverse, the simpler life forms coming first? The odds to have a proteome for Pelagibacter with 1,350 proteins would be roughly 10 to the 700,000th of power, and Raphidiopsis 10 to the 1,250,000th of power. So you are saying there is a chance that this could happen by unguided evidence? It is vastly more likely that the functional proteome, both Raphidiopsis and Pelagibacter, would have arisen by the direction of an intelligent designer, rather than by unguided random mechanisms. Now let's come back to the paper which I mentioned at the beginning. They try to make some claims and solving this riddle. And one of the claims is this: the actual identity of most of the amino acids in a protein is irrelevant. An example in nature could be the prokaryotic DNA methyltransferases, which each contain a target recognition domain, TRD, of approximately 150 amino acids that recognize specific DNA sequences, usually 3 to 6 base pairs in length, and a conserved catalytic domain. The thousands of known TRD sequences show negligible amino acid sequence conservation, despite the rather limited number of nucleotide sequences they are required to recognize. Now, the fact that this protein family does have negligible amino acid sequence conservation cannot be utilized for an inductive argument, generalizing and extrapolating. That is, that it is also the case in other protein families. We are talking here, in our case, about the undirected stochastic polymerization of the first minimal proteome. A minimal size of each protein is necessary to be functional. It could be claimed that small peptides performed some kind of job, then grew larger until acquiring a new function. But what about the protein complexes which are like machines and depend on the assembly of several subunits, like ribosomes, which have also various active sites? Such a bottom-up scenario is an entirely unwarranted claim, not supported by any scientific evidence whatsoever. And what about the 30% of proteins which only achieve their functional folds when assisted by Groel chaperones, which help them folding correctly? The next claim: as an extreme method to reduce the size of sequence space, Dill suggested that only two types of amino acid were needed to form a protein structure, hydrophilic and hydrophobic, and that furthermore, it was critical to define only the surface of the protein. These two suggestions reduced the size of sequence space to 2 to the 100th power and to the 33rd power, respectively. Now, this is a nonsensical claim entirely. First of all, there was no selection mechanism extant on the early Earth to select 20 amino acids. And even if it were true that only 5 amino acids or so would have formed the first proteins, how would the genetic code have evolved from assigning 5 to 20 amino acids in the future? The amino acid set had to be selected right from the start and be assigned to the genetic code. And the next claim, hence we hope that our calculation will also rule out any possible use of this big numbers game to provide a justification for postulating divine intervention. They claim that in their conclusion. And the response is, the explanations provided in this paper are entirely made up and based on unwarranted assumptions. There are good reasons to believe that cells cannot be the product of a bottom-up, stepwise, gradual development by chemical evolution. And Wilhelm Huck, he said a working cell is more than the sum of its parts. A functioning cell must be entirely correct at once in all its complexity. We can calculate the odds that the minimal functional proteome would arise by unguided random natural events. But we would have to assume all the 21 points listed above as a given, and there is no reason to do so. Some, if not most, are insurmountable problems, long-standing and never solved, like the chirality problem.